our Taiwanese colleagues here watching us, and it's very honor, and also I'm happy to see that the collaboration between and Changgen can extend further, and this is our third webinar, and now we move into a basic side, or probably today's topic is more translations, research, collaboration, and share ideas. I think this is a good direction. I think not only we want to extend collaboration in clinical, but in scientific research and interaction is also very important. So I hope today's webinar will be very successful, and everyone can learn and share each other idea and enjoy the meeting. Thank you.
Thanks, Prof. Pang. You're absolutely right that this is really nice to continue having a multiple different topics and a sharing session. This is really a great for many future collaborations. So I would begin the sections by little notes to the audience joining us through Zoom platform, as well as we are now on live via Faculty of Medicine YouTube webpage as well. So those audience would like to raise some question to the speakers. So because today we have two speakers, guest speakers, we'll be having the question and answer section right after the speaker deliver the talk. So please feel free to post your questions to the either platform. I'll be happy to take it, and then we'll discuss that later. Thank you so much. With that, I would like to introduce, it's a very honor to have Professor Yvonne Lim Ai Lian to join us today as one of the guest speaker. Prof. Yvonne, no, I mean, it's kind of like for us, it's like, no, she doesn't need the introduction because she's really famous for her work. And currently, I would like to mention also, currently she's serving as the Director of International Relations Office at University of Malaya. And Prof. Lim is a world-class researcher who's focusing on host-parasite interaction, epidemiology, and control of neglected tropical diseases, especially at the underserved and also indigenous community. Her work has been really well-known and also been published in many prestigious journals as well. So today, Prof. Lim will talk about demystifying the secret of our ancient foes, gut microbiota. Prof. Lim, welcome. Prof. Yvonne, you need to unmute your mic.
All right. Sorry. Yeah. Thank you so much, Dr. Selvi. It's indeed a great honor to be here. Good day, everybody. And I'm very happy and privileged to be in the presence of Prof. Pang. We have Prof. Kao, as well as Prof. Yang, who will be presenting later. Yes, so University of Malaya and Changkong Memorial Hospital has had a few series, and I'm really glad as the International Relations Director to hear of this very productive collaboration. So without further ado, just let me share my slides here. As what Selvi was saying, I will be presenting to you on gut worms. Yeah, and so how to demystify the secrets of our ancient foe through gut microbiota. Now, why do I say that worms are ancient foes? So this is because worms have been already written 2,500 years ago. If we look at the writings of Hippocrates, and he has already mentioned about worms. And recently in 2017, there was the first archival evidence of worm found in the feces of prehistoric burial grounds. So this illustrates how long worms have been co-evolving with humans. So gut worms, as we know, so this is the one that I'm showing you in the picture. This is the round worm. They basically look quite like the strength of the spaghetti. So they are parasites. And so parasites are organisms that live in or on an organism of another species. So they can live within the host or external part of the host. Now, regardless where, they are dependent on their host for survival, obtaining shelter as well as nutrient. Now, they usually do not kill the host because they need the host for their survival. And that's why they are termed as parasites, because basically they are dependent on their host for their own survival, especially in terms of nutrition supply. So gutworms have to co-evolve together with humans. And as I've mentioned earlier on, it has been happening for several million years. Now, to ensure the survival, as we know, any foreign body that enters our human body, our immunity will be targeting the foreign body. So that's why the worms over the years, they have developed mechanisms so that they can suppress the severity of diseases. And how do they do this? Of course, by immunoregulation. Now, going back a little bit about the worms, in the human gut, there is this three very common gut worms. We have the Trichuris trichura, we call them the whipworm, Ascaris lumbricoides, this is the roundworm. We also have the hookworm. There are two types of species that commonly infect humans, the Nicetia americanus and Isoclostoma duodenale. So these worms commonly manifest in terms of a patient or a host having diarrhea. Sometimes they can be anemic, there is abdominal pain and weight loss. In certain instances, when there's heavy infection or chronic infection, for Ascaris lumbricoides, because it's huge, they can cause intestinal obstruction, because sometimes if it's a very high infection, high load, there can be hundreds of worms that is living in our intestine. And when there's too many, ectopic migration can also occur. For Trichuris trichura, it can cause rectal prolapse. And for hookworm, as we know, hookworm will basically absorb the iron that is in the host, and hence it may cause anemia or pallor. Now, there are complications because of these infections, and most of the time when we look into communities that have high level or prevalence of worm infections, they have retardation in their growth, malnutrition, micronutrition deficiencies, and over the course of time, it can impair their learning and school performances. This is very much prevalent in the community, saying, communities in Malaysia. And hence it's a sort of a vicious cycle. They have reduced working capacity and then poor economic productivity. So it's very much related to poverty, to low sanitation. Now, in that sense, many of us would have presumed that worms are bad because of how it's illustrated in many of these worm infections and consequences. And now we know that gut, in our human gut, worms do not live alone. They're not there without any other co-inhabitants. So in our gut, there are other things that are also growing together and living there. So worms have to cohabitate, have to learn how to live with other organisms. And as we know, in our intestine, there's lots of bacteria. Yeah? So the bacteria that resides in our human intestinal tract are known as the gut microbiota. And they play a very critical role in regulating our innate and adaptive immune homeostasis. Now, we know that our gut microbiota consists of trillions of bacteria, and currently there's about 1,000 species of them. Now, they are very critical to our human health, and sometimes they say, you know, when you have a healthy gut, you have a healthy body. So the role of this gut microbiota is the good bacteria is to inhabit the pathogenic ones. They also play a role to metabolize our food, in terms of vitamins and hormones production, immune development and regulation, and very importantly, they help to maintain the integrity of our gut mucosa. So among the various types of bacteria or the phyla, the most abundant would be the Firmicutes, the Bacteroides, Proteobacteria, and Actinobacteria. Now, since worms and bacteria live in the gut, I'm very sure that they need to communicate, they need to talk, they need to collaborate, they need to have partnerships so that they can live in synergy. And how do they do that? What kind of things happens between them? So our team were very intrigued about this. And together with the collaborators from New York University at that time, we started this project, and it was published in PLOS NTD. Helminth colonization is associated with increased diversity of gut microbiota. So I'll just share with you about some of the results of this study. Now, so very briefly, we had detection of the parasite. Once a sample is collected, we detected the parasites, we analyzed the gut microbiota, and we did bioinformatic analysis. So this is a cross-sectional study. Among 51 participants that we had, many of them had worm infection. Almost 55% had trichuris or the whipworm, 41% had ascaris, and about 10% had hookworms. So taken all together, there's about 71% that were positive and about 30% that were negative. So what we did was to then profile the types of bacteria that they had, and we find that this about 56% had Firmicutes, 24% Bacteroides, and about 10% Proteobacteria. Now, what we could also see when we did a comparison between those that were positive for helminths or worm versus those that were negative for worms, we saw that there was a significant difference in terms of the composition of gut microbiota between these two groups. We can see that those that were positive had greater diversity in their gut microbiota, and it was significantly different. So what I'd like to illustrate, I have a picture of one of my PhD students. She likes to illustrate as gut microbiota diversity as a garden that is really thriving. And as we know, it may not be a very accurate analogy, but if you think about worms in your garden soil, we do know that some, with the presence of some worms, it does make our garden more fertile. So that is how it goes. With some amount of infection from the worms can really diversify the type of bacteria that is in your gut. And without it, sometimes it can be very monotonous. So now back to our question about, this association about worms, about gut microbiota. So we do see that there's some form of association. Can we apply that knowledge now? Can this association be used to treat certain diseases? Yeah. So that was something that was really making us think about, that's why they say a little bit of knowledge is dangerous in a very good way, because it really drives you to think more, to ask more. So we were looking at some of the data about epidemiology, very broad-based epidemiology, big studies. And we noted that the distribution of autoimmune disorders and helminths are actually inverse. And the next question is this a coincidence, or is there is a basis, a scientific basis behind this? Now, that moved us to another part of a study, and the results of that study, we got it published in Science in 2016. So how we did was that we had two components of the study. There was an animal study. One of our collaborators had the NOT2 deficient mice, whereby he knocked down certain genes, and the mice had intestinal abnormalities. Yeah. So basically, we are simulating the inflammatory bowel disease. So there's abnormal globate cells, depletion of mucus layer, and in this mice, we found that there is an increase of certain group of bacteria, which is the Bacteroides vulgatus. Now, so what we did was to infect these mice with a species of worm, and that's the Trichuris muris. So that's the similar version of the human Trichuris trichura, but muris are normally can be infected into rodents. So when they are infected, we saw that over time there was a restoration of small intestinal globate cells, and the mucus layer of this mice improves over time. Now, not just that, we also saw that there was intestinal bleeding was much less, and there was not so much of the exaggeration of weight loss, mucus depletion, and splenomegaly. So with the presence of worms in these mice, it does reduce some specific pathologies such as abscesses, epithelial hyperplasia, villus blunting, as well as immune infiltrates. So what we did was to also discover to see whether how it affected the gut microbiome profile. And we noted that before the worm infection, they mainly had many bacteria that is coming from the order of Bacteroides. And when the infection of worm was initiated into these mice, we saw that the reduction of this group, however, there was an increase, a significant increase then in the order of Clostridiales. So that was what we found in the mice. Now, we also saw that the expansion of this particular group of bacteria, the order of Clostridiales, was basically initiated, or it was associated with interleukin 13 and interleukin 4. Now, we wanted to see whether what was happening in the mouse model or the mice model was the same as in human. And as we know, in animal models, you can infect with worms. However, in the human, you can't do that. So we had to think of a way how to understand this association. So what we did was the rather reverse of it. We had to look into communities that had the infection, and then we treated them. Yeah, so looking at how it works reversely. And for this particular study, we basically did a longitudinal study as compared to the first study that was cross-sectional. Now, so when we deworm the community of, or the people, or the individual that had the infection, we realized that those that were those that before they were dewormed and after they dewormed, there was significant difference. There was a reduction, significantly reduction in terms of the richness post-treatment. So as you can see in this box, basically greater evenness and greater species richness was gotten at pre-treatment period. Now, this is just another illustration to tell you the type of abundance of bacteria that is in the pre-treatment Helminth or worm positive subjects or participants. So in this longitudinal treatment study, we saw that it was consistent with our cross-sectional study, as well as it was consistent to our animal model. So Clostridiales was the most significantly reduced order when there is deworming, when we sort of reduce the worm burden in the individual, whereas Bacteroides was significantly expanded after treatment. So what was happening in the human was very similar to what was happening in the mouse model. And this is our postulation that with the infection of the worm, so this drives certain interleukins to be expressed, and they will drive the production of interleukin 4, interleukin 13 especially, to then create a very conducive environment whereby there's increase of mucus, global cell hyperplasia for good bacteria to thrive. And the good bacteria in this case is our Clostridia. And when the good bacteria can thrive, it's just like an ecology concept. When you have many good individuals, species, then they tend to suppress the bad species. So hence there's a reduction of bacteroides strains in the human gut. So hence both the studies that was done over a period of time shows us that there's a synergistic effect with regards to Helminth infection and how it affects our immune system, and then how it's manifested in the gut diversity of gut microbiota. Now, so does that mean that there's a potential to use Helminth as a therapy? I think at this point maybe it's not so palatable. So we do need proxies or surrogates, and hence I think understanding how infections with worm influences the kind of diversity of bacteria, species of bacteria. It's with the worms and with the presence of other organisms is very, very important to be understood. So hence, we are actually in this phase now. We have gotten an NIH grant together, our collaborators, now together with our collaborators at NIH as well, to understand this better, because I think there's a lot of factors that are interplaying in our gut. It's not just the presence of the microorganisms, but there's also the influences of the micronutrients of our diet. And very recently in 2019, we published in PLOS Pathogen, and I'll pick some of the key findings that there are influences of micronutrients as well in terms of, onto our regulation of our immune system, as well as the profile or diversity of gut microbiota. However, in this instance, we saw that the worm infection plays a bigger role than the dietary intake. Yeah. In this particular study, we saw there were significant influences in terms of serum zinc and iron levels. So we want to understand this a bit more, because we also do understand with some of this worm infection, it does affect on some of the micronutrients. So it's a big challenge. It's not so easy to understand, but we do know that all these are being interplayed in our gut, and it is influencing how our immune system is responding to all this environment, the situation that is in the gut. So back to the worms. Do we think worms is a panacea, is a magic bullet? Well, it's too early to basically mention. There's still a lot more to go. There are lots of literature now to illustrate the potential of protective effects of gut worms in terms of allergy-related diseases, in terms of multiple sclerosis, rheumatoid arthritis, diabetes, even cardiovascular disease. So mechanisms, it's important to understand the mechanism. Probably we do see a certain association, but we need to deep dive into what is really happening. Now, so there are also references that talks about the ability of worms or helminths to alter metabolic status via type 2 immune response in obesity and metabolic syndrome. Now, so we are working this with Dr. Peng Lok from NIH to unlock many more secrets of the worms. And we're also working with Massachusetts Institute of Technology, MIT, to understand certain aspects of the gut microbiota as well. So with this, I hope that I have, to some extent, demystify the worms, that they are not totally bad. They can be good as well. In some instances, small doses of worms can be our friends, but of course, high doses of worms can be our foe. So with that, like I said, the demystification of the secrets of worms, thanks to advanced technology, we are able to do that. I think previously we have depended a lot on microscopy and advanced technology like next generation sequencing has enabled us to really decipher all these things. So our discovery continues with many other aspects. These are the students that are involved in the various studies. And I'd like to thank my funders and sponsors, as well as my collaborators and my students. Thank you so much. And I hope that I have sort of make you realize that worms are not so bad after all. Thank you so much. Thanks, Selvi. Over to you.
Thank you.
Thanks. Thank you so much, Prof. Iwan. Yes, you have convinced us, and we heard you many times before, and you definitely never fail to convince us on that key messages. And it's very interesting as well. I mean, you show us the synergistic effects between the immune systems and gut microbiota and the worm, the Helminth. So that is very nice and impactful work. I do have some questions, but I would like to invite colleagues from Cheng Memorial Hospital if you have any questions.
Well, I just want to say one word to Professor Yvonne. In Chinese, we say, you turn feces into gold. So you understand that you turn the feces into gold. So you can go out of the feces. So it's really congratulate to your excellent work, and really, I learned a lot today. And now we are not so afraid of the worm, but still a lot of things to do and to dig in and to find out what really happens in our guts when the worm is there. So hopefully, with your research, we can really, like you say, we can bring some significant change or maybe treatment to the clinic. So looking forward to your excellent work, and we also have a microbiota center here now. I just set up last year. So we hope that in the future we can have you visit us, and then can share together our data, and maybe we can do something good, do some good research together. Yeah, thank you.
Thank you so much. I will be delighted so I'll be waiting for the borders to be open, and I think Taiwan will be the first destination. So really looking forward. And I agree to what you say. I used to tell people that feces is something that we discard, but for the parasitologists, is like what you say, is gold to us, is treasure for us, because there's so many things to uncover. Yeah.
Thanks, Prof. Pang. Any other colleagues from Cheng Kung Memorial Hospital to share your questions, your remarks?
So according to the data, is very interesting that after the treatment, seems that the Clostridium decrease and the bacteroid increase, and that is both related to IL-4 and IL-13 increase. So do you think that this kind of phenomena is related to autoimmune disease or protection for allergy?
So as we know, interleukin-4 and 13, they are involved in the protection of anti-helminthic immunity. So definitely there is a role here. And so that's why I think they sort of play a dual role in terms of Helminth infections, as well as providing some form of association with autoimmune disease as well. Now, what we do not know is the molecular and cellular mechanism of what really is happening, and whether the two interleukins basically works in synergy. They need to work together, or sometimes, or there is an overlapping role. We do know in certain worm infections, apparently, they do not need to have both to have a protective effect. So this is, and the thing is that with one worm infection, it doesn't apply to another worm infection. So the species type of how it influences how this interleukin manifest or express and influence the immune system also needs to be understood, because it cannot be generalized. So it's a very interesting mystery, but I think this is where I think we really need to really tease them apart and understand. And if we could understand this really well, I think then, in terms of the therapy to use worm infection therapy, we do not need the worm itself, but we can see whether any particular proteins that is basically playing a major role here to initiate that kind of outcomes. So hopefully we'll reach there, but it's still in a very, it's a long way. And I think that's where I think collaborations and working with each other can fasten this discovery. And hopefully it's for the world. I think we have certain parts of the world that are very much infected by worm infections, but we have other parts of the world that are then facing many like asthma cases, autoimmune. So I think we need to study this. It's very important to understand the association. Yeah.
Thank you so much. This is very interesting story and new window for the possible cure of some autoimmune disease or other. Thank you so much.
Yeah.
Thanks, Prof. Yang. So it's very interesting. I have a few questions, but I will save it for later for Prof. Yvonne when I meet her to discuss in person. But it's nice to see how these basic science and understanding can really go into the intervention aspects. And I'm really looking forward as well to see how the story come along in terms of how that we can use this data and to drive the therapeutic intervention for many other conditions as well. Thank you so much, Prof. Yvonne. It's really a great honor. Again, I'd like to thank on behalf of Faculty of Medicine for joining us and for this sharing session. You have a nice day, and we will be continuing with the seminar today.
Yeah, thank you so much. I have to excuse myself. So Prof. Yang, you'll be presenting. I will be looking at the recording, and I hope to then connect with you via email. Thank you so much, everyone, and do have a nice day.
Thanks, Prof. Yvonne. So I would like to pass the moderation over to Prof. Cole.
Okay. Hi, good morning, colleague in University Malaya and Chang'an Memorial Hospital. It's my honor to introduce Dr. Yang Guangyu. Dr. Yang is graduated from Taipei Medical University and trained in Chang'an Memorial Hospital for internal medicine and nephrology. He has his PhD training in the Johns Hopkins University in the United States for immunology. Dr. Yang is an active researcher in immunology and has published high-impact research in prestigious journals such as Immunity. And recently, he also led the advanced immunology lab in Chang'an Memorial Hospital. Today, Dr. Yang will share with us his research about high-dimensional immunophenotyping approach for autoimmune and aging. Welcome, Dr. Yang.
Thanks for the introduction of Professor Ward, and thanks for the Professor Resnick. It's my great honor to present immunophenotyping approach for autoimmune and aging in this seminar. So the first, in today's work, we just recently established advanced in large laboratory. And today's work, we will have 3 parts. And first is how the immunophenotyping influence the research. And the second one is systemic lupus erythematous. And the third one is chronic kidney disease. When I was in Johns Hopkins in 2012, our lab published 2 paper, PD-1 and PD-L1 to treat cancer and published on NEJM. And since then, more and more people believe that, yes, cancer immunotherapy is kind of option for cancer therapy. And since then, science and nature, they alternatively choose cancer immunotherapy as the breakthrough of the year. And I believe that the paradigm has been shifted. And because in 2017, when I met my previous mentor in Hopkins, he said that there are already 60 to 70 people be treated by immunocheckpoint therapy. And nowadays, I think the percentage has much been increased. And more than cancer, I think that a lot of the other disease, their mechanism is also associated with immune response. And since Obama provided the precision medicine, many clinical doctors have tried to point out different kind of clinical markers for precision medicine, like DNA, RNA, and protein. We usually can use next generation sequencing or single cell RNA sequencing to provide a good RNA or DNA marker. However, the protein is always a problem. And we think that high parameter immunophenotyping is a kind of good model to provide the protein level biomarker related to immune response. Why are we trying to use multiple parameter immunophenotyping? According to the basic knowledge, if we have 2 color staining, for example, like CD4 or CD8, we will have CD4, CD8 negative, CD4 positive, CD8 positive, and double positive. So 2 color, we totally will have 4 combination of the immune marker. If we have 3 color, we totally will have possibly 8 combination. So it means that uncolor staining, we possibly will have more cell for analysis. For example, like 30 color, if we have 10 color and 3 triplicates, we totally will have a combination of 1024 times 3. However, if we have totally 30 colors, our combination will have 1 billion. So recently, we're trying to use this kind of combination to see systemic lupus erythematous. So this is a kidney analysis, the total analysis and analysis of normal and the patient with SLE. And during this, you can find that the orange color is a little bit increased, and this is CD8 T cell. And the blue color is increased. So that is plasma cell. So the data summary also shows that the plasma cell increased and CD8 cell increased. Did they see the data? Your face. And however, the difference is not so significant. So we further analyze the subset of CD4 T cell. So this is total CD4 T cell. And among these, we can find that the red dot increased significantly in patients with SLE. And this means the T effector memory cell. So the summary shows that effector memory CD4 T cell increase in patients with SLE. And CD8 T cell, their total number is increased in patients with SLE. However, the other subsets did not show a significant difference. And also in the B cell part, the non-switched B cell decreased in patients with SLE, the non-switched B cell. So very possibly it may be due to the increase of switched B cell, because the lupus nephritis patient will have more active and class switch B cells, so they can have more plasma cell to produce more autoantibody. And in the monocyte, the non-classical, the orange part, the non-classical monocyte decrease in patient with SLE. This is non-classical monocyte summary is decreased in patient with SLE. And according to previous review from annual review of immunology, non-classical monocytes may be represented as a kind of anti-inflammatory response. And in addition to the current immunophenotyping, I think that we can further analyze more immune cell difference. And one is the possibility of T4 helper cell. T4 helper cell is a new CD4 T cell subset, and that was established around 2010. This kind of CD4 T cell is associated with germ center activation. So they can activate B cell into memory B cell and also plasma cell that leads to more antibody secretion. And recently we collect some SLE patient and also compare with a database that comes out a mere 792 could be a micron A that is increased in patient with SLE. So as compared with normal, we can find that SLE patients have higher micron A in both urine and blood. So in order to verified the response and result of micron A, we did a CD4 T cell specific miR-1792 knockout to see what will happen in the T helper cell. And we also use the OVA alone T follicular helper cell induction. So after induction, normally we will have a significant T helper cell population. However, in the knockout mice, the T helper cell decrease. And that can be rescued by P10 over expression. And this kind of phenomena also correlates with germ center B cell. So significantly, the miR-1792 knockout will not only decrease T helper cell, but also germ center B cell. So we can suppose that the B cell decrease will associate with decrease autoantibody and decrease activity of lupus nephritis. So this kind of phenomena, we are very excited because that is consistent with our previous publication in Cell, because the HIF will increase miR-1792. And our previous publication showed that the HIF1 will increase autoimmunity. So it is correlates with miR-1792 will increase T helper cells. So we may mitigate the lupus nephritis by miR-1792 knockout. So in the knockout mice, in addition to decrease of miR-1792, we also find that B cell decrease and also plasma cell decrease and antibody decrease. And in the kidney, we can find that the glomerulus proliferation decrease and also the IgG deposition decrease. So that present that the lupus nephritis really is mitigated by a miR-1792 knockout. So the previous summary is in a patient with SLE, we can find decreased CD4 T cell and also increase CD4 memory effector cell population. So that is clearly related to lupus activity. And also we find increased non-classical monocytes. And in the future, we may do further evaluation regarding T helper cell and micron A in patients with SLE and control people. And the second target is about chronic kidney disease. Before we look into the data, we can think about recently the COVID-19, this kind of pandemic has influenced a lot of people in the world. And everyone knows that a patient with chronic lung disease, chronic kidney disease, diabetes, or age is apt to have morbidity and increased mortality when they are infected with COVID-19. So it seems that the common phenomena or common problem of this population is that their immune response is decreased. And come back to the chronic kidney disease. Previously, some people have found that patients with chronic kidney disease, they are more easy to get infection. So immunosenescence in patients with chronic kidney disease have been proposed. But what kind of immune cell have problem and the intensity and the phenotype is not so clear. So right now, we have some patients with different kind of stage chronic kidney disease, like CKD stage 3 and CKD stage 5. And the further immunophenotyping shows that the B cell decrease in chronic kidney disease, and CD4 T cell seems increase, and DC increase in this kind of population. And most significantly is the immune senescence marker like CD57, PD1, and KRAS1 increase in this population. So in the kidney analysis, this normal population is the patient with chronic kidney disease. And we can find the CD4 T cells is a CD4 T cell. It seems that the CD4 T cells significantly decrease in CKD, especially in patients with CKD stage 5. And when we look into the CD57 cell, that is cell related with the aging immune response. So we can see that compared with control, the CKD stage 3 and 5 patients have increase of this population. And PD1 is recently synced as an immune checkpoint. They are related with immune exhaustion or decreased immune response. So clearly we can find that patients with CKD have increased PD1 expression. And KRAS1 is also a little bit increased in patients with CKD. So the previous summary is that patients with chronic kidney disease, they shows increased immune senescence markers such as CD57, PD1, and KRAS1. Thanks so much for your attention.
Okay. Then Dr. Yang to share his research using the high-dimensional immunophenotyping and to understand the pathogenesis of autoimmune disease such as lupus and in aging and chronic kidney disease. So I would like to see any question from Malaya University.
Thank you so much, Prof. Yang. That was a very nice sharings on, especially on the two different disease conditions, and then your work on it with the immunophenotyping work to start with. So I wonder what's your plan in terms of the microRNA 17 goes into the therapeutic intervention, and maybe before that, is there any findings from your groups or maybe from elsewhere? What are the signaling pathway target of this MIR-17?
Thank you so much. This is a very good question. We have found majorly two targets for the MIR-1792. And one is what I showed, PTEN, because MIR-1792 can target PTEN, and PTEN decrease will increase the mTOR AKT pathway. So that will lead to not only T4 helper cell, but also increase Th1 response. So both of them will increase the immune response. The Th1 is a crucial CD4 T cell for cell-related response, and T4 helper cell is crucial for antibody humoral response. And in addition to PTEN, this microRNA also target on some regulatory T cell cofactors like EOS, IL-4, and so that our previous publication in immunity shows that overexpression of MIR-17 will decrease Treg response. So you can think that the Treg ability decrease so that the patient will only will be able to have autoimmune disease. And also in the animal model, we shows that the miR-1792 knockout in Treg cell can suppress the immune response. So it seems that this microRNA influence a lot of CD4 T cell subsets for the immune response.
All right, so yeah, so the aspects on the P10 and so the mTOR is the downstream pathway. So because we're also working on a stem cell aging and cancer, so we found some of these microRNA are the commonly deregulated microRNA, especially in a cancer stem cell and also the stem cell aging. So the I can't remember on top of my mind whether we picked up the microRNA 17, but it seems like mTOR pathway, P10, those are the pathway, that common pathway that get deregulated. I can see how that immune system could have been affected with that deregulation of these microRNA 17. So do you have any plans along the side on intervention plan based on the microRNA 17?
Yes. We recently will submit a paper related to the miR-1792 knockouts in lupus model, and we already have some anti-miR data in the lupus mite. It seems that the anti-miR-17 can mitigate lupus nephritis. And we are trying to see the next step to go to the preclinical analysis to see if that can be used in clinical. Since we already find out lupus patients have higher miR-1792. So if they are one of the mechanisms, the miR-17 knockouts maybe can provide benefit for some of our patients.
Sure. We do have questions from our colleague here, Dr. Rina. She's asking, do you think miR-17 has a broader impact in modifying an individual response to viral infection, for example, COVID-19?
This is a very good question. Since miR-17, they can increase the T-cell helper cell population. So maybe the miR-17, maybe a patient with good response to COVID-19 response will have higher miR-17 expression.
All right. Because she believed, because of the impact of the downstream, the Th1 response, so it could have been a broader application. So that would be interesting to see what's going on, especially with the COVID period or pandemic situations now. I don't see any question from this, the forum, the Zoom platform. So I do wonder, very technical or very minor things on the, when you say unswitched B cells and switched B cells, what is that? I'm sorry, I'm not an immunologist, so I'm probably want to enlighten on that.
We think that non-class switch B cell, we can find that the non-class switch B cell is decreased in patients with SLE. So I think one of the explanation is that the lupus patient have more active B cell. So most of the B cell already have class switched. So they can more easily to produce antibody. So that may be the reason.
Thank you for that. I think there's no other further questions from our side as well, I can see, but it's really a wonderful sharing section. Thank you so much, Prof. Yang, for this today's section. And I would like to call upon a closing for today's seminar. And for the audiences who are watching live live YouTube as well as Zoom, we are having a plan continue this webinar for 2021. With that, please stay tuned to have a more webinar sharing on the different topics, and feel free to put up your topic of interest as well if you would like to hear from both institutions. Right, I would like to ask maybe Prof. Ong and Prof. Pang and Prof. Kuo to have any closing remarks.
No, I think I share the view of Professor Shive. mentioned, I think we should continue this kind of webinar activity next year as well. And I hope that the audience can provide the topic that you are interested, and then we can try to fulfill your request. And I'm sure that through this kind of activity, we can know each other better, and I'm sure that we can learn from each other and work together to solve the medical problem or the unmet need in clinical. Thank you.
Yeah, thank you, everyone from Chang Kung Memorial Hospital. So I think we learned a bit today. I think this activity for me is like we plant seeds and hope some of these seeds will grow into trees, big trees, small trees. So hopefully this will progress in the future. So to everyone in UNC Malaya and Chang Kung Memorial Hospital, we hope that we will have a better year next year in 2021. So Happy New Year. Happy New Year.
Happy New Year. Happy New Year.
Thanks. So yeah, on behalf of Faculty of Medicine at the University of Malaya and also Malaysia, we wish everyone, all the colleagues at the Chang Kung Memorial Hospital, a very blessed 2021. And we hope to catch up again for next meeting, which is coming soon. Thanks, everyone.
Thank you.
Bye.
Thank you.
Bye.